So last time we went as far as talking about normal vision, 20/20, nearsightedness, farsightedness, what constituted 2200 blind. So what we're going to move on into now is the actual physiology of vision, meaning how does the photoreceptors work? How are they activated by light? So we talked about having two types of photoreceptors, the rods and the cones. The rods are a lot more abundant than the cones. We have over 100 million rods versus 6 million cones. So there's a lot more of the rods. Rods are essentially the photoreceptors that are all over the retina, whereas cones are exclusively found in the macula lutea. Sometimes they describe rods as being for black and white vision versus cones being color vision, which is true, but that's not their only difference. The physiology of photoreceptors is studied by looking at the rods, and the cones will act in a very similar manner. So one thing to notice is that the photoreceptors are physically in contact with the cells of the pigmented layer. And we mentioned before that is because the cells of the pigmented layer store vitamin A, which will be used by the photoreceptors to make a pigment that is light sensitive. This pigment is actually called retinol, and retinol is put together by being attached to a protein called opsin. And so the combination of retinol and opsin creates a rhodopsin molecule. So the rhodopsin molecule is this entire purple thing right here. And the ribbon-like structure inside is the opsin. The red molecule right here is the retinol. These rhodopsin molecules are found on a segment of the photoreceptor that is adjacent to the pigmented cells. That part of the photoreceptors, called the outer segment, is loaded with these discs made of membrane. So you can see here phospholipids, which are making up these discs, and inserted into these phospholipids are millions of these rhodopsin molecules, each one with a retinol and an opsin. The actual molecule that responds to light is retinol. And what happens to retinol is that before light hits the molecule is made by these rings of carbon atoms with this tail of carbon atoms. And because these atoms are double bonded, and I'm trying to explain these. Part of it is, oh yeah, there we go. And these hydrogens right here are close to each other. Those hydrogens and the double bonds make the tail, this tail of carbons kink like that. Okay, this is what we call the cis-retinol conformation. When light hits retinol, it's going to break these double bonds between carbons right there, and it's going to rearrange the molecule so that now it's going to it breaks the bond and weakens it, so that now the molecule can straighten out, and this is what we call the transretinol. So light is going to change that double bond so that now the tail and the line of carbons can now come down and straighten out and form transretinol. When that happens, because retinol is in contact with opsin, as retinol moves down, it's going to move the opsin protein. And when it moves the opsin protein, the opsin changes its shape, and it puts in place a series of reactions that are going to end up with the closing of sodium hits. And I'll explain this here in just a second. So when light hits retinol, cis retinol, and changes it into trans retinol, a series of events begin that is going to affect opsin, and opsin in turn will affect a series of enzymes that are going to end in the closing of sodium gates. All right, so that's essentially what light does. That's how the photoreceptor reacts to light. So what you need to know, definitely know the components of rhodopsin, opsin, and retinol. Know that retinol is the actual pigment that absorbs light, and it changes when light hits it from cis position to trans position. Cones are going to act very much the same way. And one thing about rods, before I move on to cones, one thing about rods is that they are extremely sensitive to light. So one photon of light can change the retinol of a rod. And they can absorb, they can be changed by any wavelength of light. So the reason why we describe rods as being adapted for reduced vision, for reduced light vision, light intensity, for less light intensity, is because they're so sensitive to light. They're responsible for peripheral vision. That's because the cones, which are the ones responsible for central vision, are located in the macula lutea. Okay, so one thing to get out of these, rods are very light sensitive. So they don't need much light to be activated. And that's what sometimes they're called, they're for night vision. Cones are different. First of all, cones there are three types of cones, and one group of cones respond better to red light, another group responds better to green light, and a third one responds better to blue light. So we have red cones, green cones, and blue cones. A combination of these three colors creates all the other colors. So that's for starters. Second, they're not as sensitive to light, so we need a lot more photons of light to activate the retinol in the rhodopsin. Okay. Okay. So it needs high light intensity to be activated, and that's why we say these are not only color, but they're also day vision, which is not quite correct, but okay. So rods can be set off by any wavelength. Cones can be set off by a specific wavelength, depending on the cone. All right, so we said that what happens when light hits retinol is that a series of events are going to be put into motion that are going to close sodium channels. So if you remember from last semester, that would make no sense, because what we said last semester is that a stimulus, which in this case it would be light, is going to cause the opening of sodium channels, which will lead to depolarization, so that the cell will be depolarized and an action potential will be created. That's how we explained it last semester. We also said that would be an excitatory postsynaptic potential. And there are instances in which neurons, instead of being excited with the stimulus, they're actually inhibited by the stimulus. So what happens with photoreceptors is that when light is not available and they're not being stimulated by light, that's when the sodium channels are open. And as the sodium channels are open, positive charges go into the cell, depolarizing the cell, creating an action potential. And the action potential causes the photoreceptor to continuously secrete a neurotransmitter called glutamate. So in the dark, what is called the dark response, when light is not activating the photoreceptor, that's when the photoreceptor is actually secreting a neurotransmitter. It's creating an action potential and secreting a neurotransmitter. Turns out glutamate is an inhibitor, which creates an inhibitory synaptic potential into the bipolar cells. So the bipolar cells are essentially shut down and there's no more communication going on. When light hits the photoreceptor, retinol is changed from the cis to the trans position, which causes the set-off of a series of events that will lead to the closing of the sodium channels. So now, all of a sudden, the cell is not polarized anymore. It's going to be hyperpolarized. It's going to begin to lose potassium and become very negative. There will be no action potentials being created, no glutamate released. So the neurotransmitter that was shutting down the bipolar cells is now gone, and bipolar cells will begin to create action potentials, be depolarized and create action potentials. So now the bipolar cell is free to begin sending messages, sending signals to the ganglionic cells. So that's essentially how the photoreceptors are going to respond. So what do you need to know from here is that in the dark response of photoreceptors is when the photoreceptor is depolarized, creating action potentials, releasing glutamate. Glutamate is an inhibitory neurotransmitter, and so there is no more signal going on into the bipolar and the ganglionic cell. You should know what light does to the receptor, to the photoreceptor. It changes retinol from cis to trans, closes sodium channels, it stops the action potentials, it stops glutamate release, and now bipolar cells can be active and depolarize and send signals to the ganglionic cells. Okay, let's just going over what we just talked about. So another characteristic of retinol is that when it changes from the cis to the trans position, which happens with light, the molecule has to be resynthesized. So it cannot just be tanked again and changed back to cis. It actually has to be remade. That requires enzymes, that requires energy, that requires time. This event in which the retinol all of a sudden is in the trans position and it cannot be activated again until it's remade, is called bleaching. So light is going to bleach retinol, meaning now it cannot work. Light hits retinol, it changes it into the trans position, now it's bleached, and it takes quite a few seconds, as long as 6 seconds or so, for the molecule to be remade, and the retinol in rhodopsin to be made into the cis position again so that it can work once more. So retinol is bleached. It's going to take time to reactivate it. So that's what accounts for the light and the dark adaptation that we experience, is this bleaching of retinol. So when you move from dark to light, so you have been in the dark for a long time, what that means, you have lots of photoreceptors ready to go. You do not have bleaching going on. So if somebody turns on the light, all of a sudden you're blinded. And that's because all of a sudden light strikes a whole bunch of photoreceptors all at once, and all of them get bleached all at once. So for a few seconds, you have literally non-working photoreceptors until they get remade again. The retinol gets remade again, and now they have again the working rhodopsin. When you're moving from light to dark, now there's another step in another adaptation. You're in the light, therefore you have bleached photoreceptors. So you don't have all of your photoreceptors ready working. You step into the dark, and you have, let's say, I don't know, just to put a number, 50% of your photoreceptors are bleached, the other 50 are okay, but you only have 50% of the photoreceptors that are okay, and all of a sudden you step into a dark place. You won't be able to see very well at first until those bleached photoreceptors begin to be active again, and now all of a sudden you may have 80% of the photoreceptors active and you can see better. So that's why when you step into a dark room, at first everything is pitch dark, but you wait a few minutes, you begin to see more and more and more and more, because you begin to have more and more photoreceptors that have been reactivated. So that explains that adaptation, what's called light and dark adaptation. So again, this is just showing how the bleaching takes place. You should know what bleaching is. And now we come to the neuronal pathways, which is the route that the visual information takes all the way from the eyes to the visual cortex in the brain. So from last semester, the visual cortex was located in the occipital lobe of the cerebrum. So the information has to travel from the eyes at the front and be taken all the way to the very back of the cerebrum. The nerve that leaves the eyeballs is cranial nerve number 2, optic nerve, and the sum of the fibers are going to crisscross from right to left at the optic chiasm, right in front of the pituitary gland. And then from there, the information travels to the thalamus, sorry, not hypothalamus, but thalamus, right here, through the lateral geniculate nucleus of the thalamus. And from the lateral geniculate nucleus of the thalamus, it goes into the optic radiations, right here, into the occipital lobe. So we have optic nerve, optic tract, after this, so optic nerve, optic chiasm, optic tracts, thalamus, more specifically the lateral geniculate nucleus of the thalamus, and from there into the visual cortex. So that's the path that the information will take to the visual cortex. Humans have binocular eyes because both eyes are at the front of the face, which means that they're both giving you a slightly different versions of the same picture, angles, slightly different angles of the same picture. And when our eyes put both pictures together, we get a 3D image. So we get depth. So animals that have their eyes on the side of their head, they do not have binocular vision. They do not have depth perception. We have depth perception because both eyes are at the front, and the combination of the two slightly different images gives us depth. Okay, abnormal conditions of the eye. Cataracts are a cloudiness of the clouding of the lens that occurs with time. So lens proteins. Diabetic retinopathy. This is the #1 cause of blindness in Western countries. This is damage to the retina due to diabetes. Glaucoma is an increase in intraocular pressure due to an increase in aqueous humor, which probably is not draining properly. And macular degeneration is the loss of the cells of the macula lutea. And because most of the cells in the macula lutea are cones, we begin to lose central vision. So peripheral vision is okay, central vision is what's lost. So you should have an idea what these conditions are and what part of the eye they affect. Yeah, go ahead, Tava.
So I was just curious. I've noticed, especially with myself, because I have horrible eyesight, that the one eye, or I'm assuming it's the brain, kind of combines the information from both eyes, and if one eye sees better than the other, that it compensates, like in your overall vision. Is it just our brain that's like, compiling that and giving the information? Like, how does that work? How's the best way to explain that?
Does it? But yes, the brain is the one that is it's learning to take the image and compensate and give you a clear image. So what you're saying is, if you were to close an eye, you will see maybe not as well with the eye that's still open, and if you close the eye that's not as good, you look through that eye and you see better. But yet, when you have both eyes opened, you have good vision. So the bad eye, the bad vision, is being compensated by the good vision. And yes, the brain absolutely does that. How does it do it? I would imagine it probably takes more into account the information coming from the good eye. Probably that eye is sending better information, and that's why that one is the one that is paid attention to. That's the one that is actually integrated. That would be one answer. Keep in mind, oftentimes people have different lenses in each eye. So they may have one eye with a lens for close vision and the other one with a lens for far vision. That way they don't have to use two different glasses. And within a matter of days, the eye learns that one eye is for close vision, the second eye is for far vision, which is crazy to think about it. But the people who use those kinds of lenses get used to them very quickly. So what really gets used to them is the brain. The brain gets used to it and it knows. And when one eye is, when you're focusing on something close, one eye takes over. And then when you're looking far, the other eye takes over.
That's a really good point with the different lenses. 'Cause my father does that. I wasn't even thinking that, and I always thought it was fascinating. Like, what are you going to do? Close one eye and then the other to see near or far? But you're right, it compensates. It's so interesting.
It is. It is very interesting, and I could probably benefit from those kinds of lenses.
You and me both.
It gives me anxiety of what my brain is going to be doing. I just can't do it. I can't take the plunge because to me, I think, oh, I'm going to confuse my brain. No, you don't confuse your brain. The brain learns. But anyway, just the thought of it seems so amazing.
I know it's weird for me too. I agree. Oh, thank you.
No problem. So that's basically it for that part. So we can move on now to hearing and balance. So we'll do the same thing we did before. Let's go ahead and start with the PowerPoints on, let me just find it real quick, on the, here we go, on lab PowerPoints. And just go over the anatomy. And actually this is not very complicated or extensive, actually. So by going through one set, we pretty much cover both.
Can I ask you one more question really quick? I'm sorry. I'm thinking about this as we're going back and forth between lab and lecture.
Yeah.
I typically last semester, I always took my lecture test before my lab. I don't know why I did that, but I did. Do you have a recommendation for one? I typically try to take them back to back because it is all the same information. It's just a little bit different way of looking at it. But if not, do you have a recommendation one before the other? Because they typically go in pairs.
Yeah, in this lab they continue to go pretty much in pairs. I think, but it seems to me as if your system is working for you, why change it? But I think that if I were to recommend, I would probably recommend lab first. If anything, because it seems to me it is a little bit easier. And once you have the anatomy in place, you have a big chunk of the lecture material in place. You can, if you want to review lecture, you can now go ahead and review the physiology, if you will, the parts that are not the anatomy. Since you already have the anatomy, you've taken the test, you've been tested on it, you've been kind of sort of like, it. And so now it's just a matter of filling in the blanks with the physiology. That's how I would approach it. Yeah.
That's probably a good point. I had a little bit of a hard time with the practical side of it last semester because the way the instructors for lab kind of spoke about everything, and because they weren't always in unison, she didn't give the physiology behind it. And the way that I absorbed the information is both, because I'm like, okay, well, I know that the cochlea is here because it does this, and that's why it looks like this. Does that make sense? So which way to do it. So I'll try it that way, and let's hopefully, I hope I could do as well. But let's see, maybe that'll help me.
Right. Try it once, and if it doesn't work, just go back to your old system.
Okay. Thank you.
I don't know. But yeah, to me, it makes more sense. And I'm trying to face classes. Typically, if we had the lab practical first, and then after that, students had a few days to take the lecture, I think they did better because sort of like they had already studied for the anatomy. Now they could just concentrate on the other part of the material. So yeah. So anyway. So when it comes to the ear, what we do with the ear, if we divide it into sections, external ear, middle ear, and inner ear. And the external ear and the external and the middle ear are exclusively for hearing. So they are the structures there are for that purpose. Whereas the inner ear is where the receptors are located. So that the inner ear is going to be for hearing and equilibrium. Okay. So what the external ear does is it collects sound, and sound are vibrations of air. When it comes to structures of the auricle, which is what we call the ear, the rim out here is the helix, the lobule is the end of the helix right there, and that's probably the only two structures that are on your list. And then from there, the purpose of the auricle is to collect the air vibrations, and those air vibrations are now going to travel into the ear, into what's called the external acoustic canal, right there. So it travels into the external acoustic canal, these air vibrations, and the external ear ends at the tympanic membrane or eardrum. So the vibrations are going to hit the tympanic membrane and make it vibrate. So, and those vibrations are now going to be transferred into the middle ear. The middle ear is made of 3 bones, corticals. The bones are malleus, incus, and stapes. Malleus is the one closest to the tympanic membrane. Stapes is the one furthest, so this will be the one closest to the inner ear, or the entrance into the inner ear, which is the oval window. There is also a tube in the middle ear called the eustachian tube, auditory tube, or pharyngotympanic tube. It has 3 names: auditory tube, eustachian tube, or pharyngotympanic tube. And this tube connects the middle ear with the nasopharynx, which is essentially the back of the nasal cavity, the throat, part of the nasal cavity. So in the back, it's not part of the nasal cavity, but it's the throat, the part of the throat that is at the back of the nasal cavity. That's called the nasopharynx. And we'll talk about the function of the eustachian tube. So the middle ear the first ossicle is the malleus, which is in physical contact with the stapes. So when the stapes vibrates, the malleus vibrates. And the malleus vibrates and transfers the vibration to the second ossicle, which is the incus, and it vibrates and it transfers the vibrations to the stapes, which is the last ossicle. And the stapes is physically on top of the oval window, which is the entrance into the inner ear. The oval window, you cannot see it because the stapes is right there on top of it. This little tube right here is the auditory tube, eustachian tube, or pharyngotympanic tube, which is connecting the middle ear to the nasopharynx. So that when air is in the middle ear, and if we go, for example, travel on a plane and we begin to ascend, the pressure is going to change so that the pressure outside is going to go lower because now we're under less air environment. The pressure in the middle ear has remained what it was when we were on earth, on ground. So we have a higher pressure inside at that point. And that's going to make us want to yawn, And when we yawn, we open the eustachian tube, and the high pressure air in the middle ear escapes into the throat and prevents that high pressure from being trapped in the middle ear and possibly harming the tympanic membrane, for example. So that's the purpose of the eustachian tube, is to equalize pressure between outside and middle ear when the pressure outside changes. So we get this urge to yawn or to chew on something or swallow, that's something else. So things that open up the eustachian tubes will be yawning, chewing, swallowing. So
That's why they always had us sucking on lollipops when we were going up and down, I'm assuming.
there you go. Yes.
Though, I used to travel a lot as a child because my father's job. I distinctly remember I had a lot of trouble with this. And I distinctly remember that flight attendants would put a warm, like a hot paper towel, like a wet hot paper towel in the bottom of a cup and pressed it against the ear. Is it the moisture or the like trapping the air? Or what would help decrease that pressure in that case?
So a warm paper towel doing
Like,
is
like a styrofoam cup almost, and then they would have me put the open side of the cup as flat, push it as hard against the sides of my head to like trap maybe the warm moisture and like that kind of. That's why I'm wondering like
what it would do. It would probably it may have the moisture may have and maybe push the air out of the eustachian tube. Yeah, it's kind of like open up because I don't know if that air would have come out in this direction.
Way.
Yeah.
I just remembered thinking that was so strange as a child. So when you're explaining this, I'm like, wonder why they did that. But it worked. So
yeah. It was just creating a what would have been some type of pressure here that would have pushed it in and push that air out.
So coming because of the pressure in the eustachian tube, or does it have anything to do with the tympanic membrane?
Tympanic membrane definitely has nerve receptors. If you were to outside, it would hurt. It has definitely nerve receptors. Yeah.
Sorry.
So that's another picture here of the tympanic membrane with the malleus. You don't see the incus very well, but we do see the stapes against the oval window. No, this funny looking thing here, that's the inner ear. Yeah, well, we're going to go into that one in just a second. And this is just something I found on the internet. That's the actual little malleus, incus, and stapes inside the inner ear. Okay, the inner ear is really where everything is happening. First of all, it is located inside the temporal bone. And if you go back to last semester, when we're looking at the temporal bone, there was a little on the interior aspect of the bone, in the inside the cranial cavity, there was a little bump there, which was called the petrous region. As a matter of fact, it was like a ridge like that, and on one side of the ridge was the internal acoustic canal, and on the other side somewhere was the carotid canal. But nonetheless, that little bump here is where the inner ear is located. So the inner ear will be into the temporal bone. That's the first thing. Second is that it is made of three components: the vestibule, the semicircular canals, and the cochlea. Vestibule and semicircular canals are housing equilibrium receptors. Cochlea is exclusively for hearing. And all of these entire structures, the vestibule, semicircular canals, and cochlea, are made of bone. And inside that bony structure, there is a space into which the same structure made of membranes is housed. So that's why they talk about the bony labyrinth and the membranous labyrinth. So this picture here is trying to depict that. So what they're showing you is that this outer area, this yellowish area here, is bone. And then there's going to be a little bit of a space between the bone and the red structure, which is made of a membrane. So exactly the same shape, but it is inside. So it's almost a bony tube, and inside the bony tube is a membranous tube. And then these tubes have these funny looking shapes, like a snail, for example. So here they're trying to depict the same thing. This outer area is made of bone. There's a little bit of a space filled with fluid, and then this pink structure that has exactly the same shape as the bone is the membrane, the membranous structure. So the whole inner ear is made in that manner. The semicircular canals are these arches here. There's 3 of those, which are going to end, if you look at them closely, they end in this enlargement right there, called the ampulla. So each one of them has this enlargement. It is in those enlargements where equilibrium receptors for dynamic equilibrium, meaning rotation, are found. So you can see the nerves right there, ready to take information from those receptors. And then those are the semicircular canals. Then you can see here these sac-looking structures right there. That's the utricle, and this will be the saccule. So we have this one right here and this one over here. Actually, that one is part of too. Those structures make up the vestibule, and the vestibule is where static equilibrium receptors are located. Static equilibrium is when we tilt the head, as in, for example, you're sitting in a car and all of a sudden the car takes off and your head goes back. The static equilibrium receptors are the ones that detect that movement of the head going back, versus twirling, for example, which that would be the semicircular canals. And then to one side, we have this snail-looking structure, which is the cochlea, that is going to house hearing receptors. The nerve that is going to carry information about all of these receptors are going to be 2 nerves, actually. One is called the vestibular nerve. The other one is going to be called the cochlear nerve. And when both these nerves exit together from the middle ear, from the inner ear, they come together into the vestibulocochlear nerve, which is cranial nerve number 8. Okay, so now it's just a matter of, again, reviewing all these structures. This is, again, something else I found in the internet, and I thought it was good at showing you the bony labyrinth versus the membranous labyrinth. So here, the 8 and the 7 will be the semicircular canals. These 2 will be the ampulla. That will be another ampulla. This is another ampulla. That's where the receptors for dynamic equilibrium are found. Number 14 is the utricle. Number 4 is the saccule. And inside those, we're going to find the receptors for static equilibrium. This structure right here is the oval window. So this is the place where the stapes is going to position itself. And then to one side, we have the cochlea, which is the place where the hearing receptors are going to be found. And again, you have 2 sets of tubes, the bony and the membranous. Now, between bony and membranous, there is a liquid called the perilymph. And then inside the membranous labyrinth, inside the membranous sac, there is another liquid called the endolymph. So there is liquid inside the membranous structure, and there's liquid outside bathing the membranous structure between the bone and the membranes. So more of the same. So it's just a matter now of identifying the structures. The vestibular ganglia are these ganglion where the bodies of the sensory neurons are found. Remember, sensory neurons were unipolar neurons, so they had that little body to one side. So those little bodies are all found in these ganglia while the dendrites are coming from here, and the axons will leave towards the outside, going to make the vestibulocochlear nerve. There's another ganglia which we cannot see here. That would be the cochlear ganglia. More of the same. This is more of a one that we have in the lab. One thing to show here is the oval window, which is the exit for the sound waves. So let me go ahead. Give me a second. There's another one. This one is better. Okay. So we said the oval window, the stapes is against the oval window. So the vibrations are going to travel from the stapes into the inside and begins to vibrate the fluid, the perilymph, eventually the endolymph inside also. Those vibrations are energy, and they have to be dissipated. They cannot just stay there vibrating forever. So what the route that the vibrations will take after they travel through the cochlea is they're going to go out through the round window, which is this little circle right here. The round window has an actual membrane on top, and the vibrations push on the membrane and dissipate the energy of the vibrations. The round window, you could think of them as the exit for the vibrations. And that's what that is right there. That's the round window. Or calls it oval window? Maybe. Looks more like the round window, but okay. The round window is completely round, oval is completely oval. Okay, so we said that in the utricle and the saccule, we have the receptors for static equilibrium. To be more specific, the receptors are going to be all clustered together into a place called the macula, and the receptors themselves are neurons called hair cells. These will be the actual receptors. There's three semicircular canals, a superior, posterior, and lateral. At the base is the ampulla, which is where we have the receptors. The receptors are again called hair cells. So far, all the receptors for equilibrium are called hair cells. In this case, the receptors are clustered in the crista ampularis of the ampulla. Okay, so that will be superior semicircular canal, posterior semicircular canal, lateral semicircular canal. So superior semicircular canal, lateral semicircular canal, and posterior semicircular canal. Okay, so again, superior, lateral, and posterior. Okay. This is an actual utricle. So you can actually utricle and ampulla. So this is the ampulla right here, and this is the crista ampularis where the receptors for dynamic equilibrium are found. Notice that the ampulla is in connection with the utricle. So this is the utricle, and here's the macula where the receptors for static equilibrium are found. So to give you an idea, we were looking at this area right here and this area right here. They're adjacent even though their receptors are different. Okay, now the cochlea. Again, the cochlea is for hearing, is where the hearing receptors are located. And the best way to think of it is going to be a tube inside another tube. So if you think of the cochlea as these long tube, so this will be the oval window, and you take that snail-looking thing and you unroll it and make it into a tube, and now you go ahead and and so the round window will be right there, and now you're going to put another tube inside, right there. Now what you have is a space right here filled with perilymph, another space down here filled with perilymph, and a space in here filled with endolymph. each one of those spaces have names. And so the first space is called the scala vestibula. The one inside the membranous tube is going to be called the scala media or cochlear duct. And then the one below is called the scala tympani. Okay. Now, if you were to take these and roll it into a snail and then cut it, you're going to end up with many of these structures. Hard to draw like that. And then another one here. And then another one here, which makes it confusing, but that's basically what's happening, is that you're going to be cutting through many of these structures all at once. So you do not need to know the scalas. I'm going to show you here in a minute a model that has all these scalas where you can actually see it. Let's see if I can have, okay. So this is the scala vestibula, which has perilymph on top. The one inside is the scala media or cochlear duct, which has endolymph, and then the scala tympani, which has perilymph, and the round window. Helicotrema, I forgot that one. That one is the tip of the whole structure. So if you had it as a tube and the other tube inside, the helicotrema was the tip of the tube right there. That's the helicotrema. That tip of the tube will be the helicotrema. So in a model, the helicotrema will be that right there. So this will be the scala tympani, obviously the scala media, and on top will be the scala vestibula. Okay, more of the same. Okay, this is the one I was looking for. All right, right here. So this is a cut through the cochlea, and it has not been unrolled. So you have a cut through several layers of this tube. So you can see here the scala vestibuli, the scala media, the scala tympani. Scala vestibula, scala media, scala tympani. And you can see them all around. Now, let's go a little bit further. Make sure I didn't leave anything out. Yeah, the cranial nerve number 8, the cochlear nerve branch of the vestibulocochlear nerve, is the one that will collect information from these areas. The ganglia there is called the spiral or cochlear ganglia. Okay. The receptors are going to be found in the scala media or cochlea, clustered into an area called the organ of Corti. This will be the equivalent of the macula or the utricle and the saccule, and the ristampularis for the ampulla. The receptors are called hair cells. So notice that hair cells are the names for the receptors for hearing and for equilibrium. And I will talk about the other structures right now here in a minute. Okay, so this funny looking thing you see here and here and here is the organ of Corti inside the scala media. The membrane that separates scala media from scala vestibuli is the vestibular membrane. So this right here is vestibular membrane. This right here is vestibular membrane. This will be vestibular membrane. The membrane that separates the scala media from tympanic membrane is the basilar membrane down here and down here. So vestibular, basilar, basilar, vestibular, basilar, vestibular. We'll get better pictures of these here in just a second. All right, that's a cochlea, an actual cochlea being cut. So that will be vestibular membrane. This is the tympanic membrane. And this right here is the cochlear membrane. And perfect. So this will be the vestibular membrane, scala tympani, sorry. Scala vestibula, scala tympani down here. This is the cochlear duct or scala media. This is the vestibular membrane. This is the basilar membrane down here. And the organ of Corti is this area right here. Okay. Now, let's look at these from a model, which is probably the best way to look at it right here. So this is a model of these structures. This is the scala vestibula right here. This is the scala media or cochlear duct. This is the scala tympani. What that means now is that this is filled with perilymph. This is filled with perilymph. The scala media is filled with endolymph. The membrane that separates the scala vestibula from medial scala media is the vestibular membrane. The membrane that separates the scala tympani from medial membrane is the basilar membrane. In the organ of Corti, which is the place where the receptors are found, this is what it looks like. So this is scala vestibuli, scala tympani, basilar membrane down here, vestibular membrane up here. And in the organ of Corti, we're going to find the hair cells, and you can see the little hair cell here, and the cilia that gives it its name, another one here, another one here. These are going to be supporting cells over here. The membrane right on top of the hair cells is called the tectorial membrane up here. So again, tectorial membrane, little hair cells right there, basilar membrane. So this is what's going to happen. Vibrations are going to come, vibrations of air, and move the perilymph. Those vibrations are going to travel down to the scala tympani and make the liquid, the perilymph, also move in the scala tympani. And that movement is going to push on the basilar membrane. And as the basilar membrane begins to move upwards, the little cilia in the hair cells begins to hit against the tectorial membrane. When that happens, the hair cells, which are neurons, are activated. So sodium channels open, or I should say, they are depolarized. Ion channels open, positive charges go in, and the cell creates an action potential. So it's that physical bending of the hair cell as they hit against the tectorial membrane is going to depolarize the hair cells. These are mechanoreceptors. So those are the structures that you should be able to identify for this lab in the inner ear. Oh, there we go. This is the ganglia, the spiral ganglia, which collects the information. So here are the bodies of the neurons, whose dendrites are going to be right below the hair cells, collecting the action or receiving the action potentials from the hair cells, and the axons will go out and form the cochlear nerve, which later will become the vestibulocochlear nerve. So that is the anatomy of it all. Now, if we switch to the physiology, we can again put it all together, hopefully a lot easier, because now you know what's going on.
Can I ask a quick question? Not terribly on the cochlea. I've noticed that depending on the textbook or the PowerPoints, and even sometimes in both, they vary. That the cranial nerves are sometimes referred to by their number and sometimes referred to by their name. Obviously, names are a lot easier because they go with the structure a lot of times. But on the test, is it reasonable to think that they'll all be by number then?
It could be either. You should know both.
Learned it. It's one thing that has not stuck well, so I need to review it again. But I just wanted to.
No, it's both, and actually, the reason why it's both is just the reason that you mentioned, is that sometimes they use the names, sometimes they use the numbers. You have to learn both, and this is one thing one piece of information that is not going away. So this is going to be in every test you're going to ever take from now on because they're so important.
So I definitely need time on that.
Yeah. Name a number. I know it'll serve you well. There's only 12 of them, but still. I know. This is said, and then I know. Yeah.
For some reason, the peripheral nerve stuck with me, but the cranial didn't. I don't know what's wrong with me, but I'll go back. I'm strange.
Yeah,
I'm probably off.
Yeah, especially because this semester is all cranial nerves.
Yeah. Okay. I'm gonna go back. Thank you.
This semester is all cranial nerves, so they're just not going away. That's all.
Yes,
but yeah. So just review anatomy of external ear, middle ear, and when it comes to functions, just to make sure I didn't leave anything. Yeah, when it comes to function, the external ear collects air vibrations. Middle ear amplifies vibrations, meaning makes them bigger, so that the mechanical movement of the ossicles actually exaggerates the vibrations. That's part of the function of the middle ear, and then transmits those vibrations into the inner ear. So know the structures. Know that tympanic membrane is the eardrum. Okay, that's talking about that. Second, amplified. There you go. So it's going to be amplified. So it's more than just passing on the vibrations. It's going to do more than that. It's going to amplify. It's going to make it exaggerate and make them bigger. Okay, this right here, pharyngotympanic tube equalizes pressure. Okay, we got that. Know the order of the ossicles. So malleus first, incus second, and stapes is the one furthest away. Know which one is touching the tympanic membrane. Know which one is against the oval window. So you cannot miss the order. Yeah. Okay, sound attenuation reflex. This is a reflex carried out by cranial nerve number 5 and cranial nerve number 7, which will be the facial nerve and the trigeminal nerve. And what they do is they're going to respond to loud sounds by innervating, by contracting the tensor tympani and the stapedius muscle. So the response is going to be to contract a muscle that stabilizes the tympanic membrane and a muscle that stabilizes the stapes, so as to prevent them from over vibrating. Okay, so this is how this reflex functions. When there is a loud sound, these nerves are going to immediately take the information and are going to be activated. Sorry, the loud sound comes from the vestibulocochlear nerve. The integration center immediately sends the message through 5 and 7 to tighten these muscles to immediately prevent overvibration and possible damage to the inner ear by the overvibrations. Immediately, the tympanic membrane and the stapes are tightened. Okay, this is the problem. The problem is that this is a slow reflex. It takes a few seconds for this whole operation to kick in. So that means that a sudden loud sound will not give time for the sound attenuation reflex to kick in, and it could potentially damage the inner ear. So that's why when you go to a gun range, for example, you wear ear protectors. It's because the sudden loud sound of a gun going off could damage the inner ears. Another problem with the reflex is that it doesn't last very long. It only lasts for 10 minutes. So any loud sound that lasts longer than 10 minutes, your ears are without protection after that. And again, that's why if you work in a place that's very loud, you will wear ear protections because the sound attenuation reflex is not going to protect you then. So know what it is, know what the downfalls of the sound attenuation reflex is. Okay. Now, inner ear, again, just know which structures are part of equilibrium, and the other one is part of hearing. Just know the structures, what we find in each structure. So we pretty much find the same pictures here. So the three scalas, know which one has perilimbs, which one has endolymph, which one has the receptors. Know that the organ of Corti is where the receptors are found within the cochlear duct. And there's the organ of Corti right here. You can see the little hair cells right there. There's the little hair cell with the little cilia, the tectorial membrane, the basilar membrane. That's an actual cochlea. Okay. Okay, just all this is anatomy. All right, so this is the actual little cilia from the hair cells. And you can see that the cilia are at different heights. And the taller ones are connected to the shorter ones by this little protein that connects to another little protein in the shorter one. So we're going to have a little close-up, I think, on the next one, the next PowerPoint. Yeah, and you can see the connection right there. This protein right here is an ion gate. Actually, I take that back. The purple one is the ion gate. There we go. That makes more sense. So that when the basilar membrane begins to push this whole structure upwards, and the structure hits against the tectorial membrane on top, that is going to move the cilia back, and the little connection between the two cilia are going to be stretched, and that's going to physically open the gate. And positive charges are going to go in. And that's what's going to depolarize the hair cell. So that's what they're called mechanoreceptors, because it's a physical opening of these channels by the movement of the cilia. Okay, that's kind of cool. If you look at and I don't really typically go into these, but the reason why I'm not calling it sodium ions, because they're actually potassium ions going in. So instead of sodium depolarizing these cells, it's actually potassium that depolarizes these cells. So, we spent last semester showing you how things are supposed to be, and we spend this semester showing you how things are never the way they're supposed to be. So anyway, often there's a lot of exceptions to the rule. So in this case, instead of sodium depolarizing the cell is potassium. These little hair cells have less potassium inside than outside. So which is exactly the reverse of what we said neurons should be last semester. The endolymph is a potassium-rich fluid. So that's why the potassium is what goes inside and depolarizes the cell. And that's a close-up view of, again, the organ of Corti. So you should have an idea of how sound travels, the structures that it travels through. The loudness of a sound has to do with how many hair cells are activated. The pitch of the sound, these are the waves, so this would be a pitch. So that when the waves are closer together, you have a higher pitch than waves farther apart. The pitch of the sound has to do with where in the cochlea, which hair cells are activated. So where in the basilar membrane, what part of the basilar membrane is stimulated? So really, ultimately, it's the basilar membrane. So what happens is that the basilar membrane is not the same throughout. It is actually, wide and flexible at the end, and narrow and stiff at the beginning, so that high frequency sounds make the end of the basilar membrane vibrate. I'm sorry, it's the opposite because this is limber, so it doesn't need as much energy. So low frequency sounds make the last part of the membrane, which is limber, it's more flexible, easier to bend, vibrate. High frequency sounds, which have more energy, and make the beginning of the membrane vibrate. So high frequency sounds at the beginning, low frequency sounds again, obviously in the middle are the other between high and low frequency. So where the vibrations are capable of making the membrane vibrate is how we're going to detect the pitch of the sound. And this will go over, you should know the path that sound waves take all the way into the cochlea. So they're going to go from the external auditory canal, they're going to hit the tympanic membrane, make the three little ossicles vibrate, go through the oval window, into the vestibular scala vestibuli. From there, they're either traveling directly down to the scala tympani or go all the way into the helicotrema and travel to the scala tympani. And as they do that, they distort the basilar membrane and activate the hair cells. They're going to be dispersed through the round window. Okay, auditory pathways, we're not going to go too much into these. It's just going towards the brain, to the auditory center via the vestibulocochlear nerve. The auditory cortex is in the temporal lobe of the brain. Okay. All right, presbyacusis is similar to presbyopia. The hair cells become non-functional over years, so we have less hair cells, and that causes
I think the teacher got locked out from the classroom, maybe.
Yeah, I think you're right. It just said she left the room. She'll be right back. This has happened before.
Yeah.
I don't know, you were in our class last semester?
What was that?
Earlier this semester? Were you in the first anatomy and physiology one just before this, or with her now?
No, I actually took anatomy and physiology #1 like a year ago.
Oh my
So this
goodness.
is all
You
kind of new
remember a
to
lot of
me.
this, I know.
No, I don't. I took it on the summer class and it was
Oh,
fast
wow.
paced. So yeah, it was tough.
Oh, I can't imagine. I mean, it's hard enough in 8 weeks. So I'm in her microbiology class. It's 16 weeks too. So I'm like super used to her teaching, and it's so much information. My goodness,
Yeah,
just
no.
I mean, all I'm taking is, well, I took ANP1, ANP2, and micro this semester, and I'm like every second of every day studying. It's so much. So
Oh my
uh,
God, good luck to you. Are you going for nursing or?
I feel bad for you too, because like that's why I was asking her about the cranial nerves. It's like I remember the names of all of them, like abducens and oculomotor and all that stuff, but I don't remember the numbers for all of them. And I bet for you, using it every day, you got to go back and read that too, huh?
Yeah, yeah, that's what I'm doing right now. I'm like trying to, that's why I'm here, like trying to remember everything, getting everything into my brain.
She's really good. Have you ever had her before for class or no?
Oh, it's actually my first time. I had Otero and Allegra, and they were pretty good too. So
She provides you with lots and lots of information, like the study guides. I take all the information from our PowerPoints and kind of filter it into my own study guide, which I kind of expand on hers. And I got 100% in her class last semester for Anatomy and Physiology 1. So she gives you all the information that you need to like succeed. It's just a matter, a matter of putting in the time, because you do need to like know the information both ways in a way. But I think she's back now. So Sarah, can you hear us?
I'm back. Yeah, my internet, I don't know what happened to it all of a sudden.
No, that happens. Thank you.
Yeah, all the time.
Really, I just, knock on wood, doesn't happen very often here, but oh well. And of course, I don't see the special census PowerPoints. They're gone. That's just great. Oh no, never mind. They're here. They're here. They're here. I don't know what I'm talking about. Okay, excellent. All right, very good. All right, so this is really not much left. Equilibrium, again, a lot of it is just anatomy and remembering which structures are static equilibrium and equilibrium. So saccule and utricle static equilibrium, and semicircular canals are the dynamic equilibrium. Remember, the cells are exactly the same. So you could take a little hair cell from the cochlea and put it in the ampulla of the semicircular canals, and they're going to begin working for equilibrium instead of hearing. So the cells are just virtually the same receptor, just in different places. They're all called hair cells. In the utricle and the saccule, they're located in a place called the macule. And the way this works is actually pretty cool. In this case, what happens is that there's this slab of jelly-like material that is filled with little rocks, calcium rocks, and it is all positioned on top of the cilia of the hair cells, so that when we tilt the head, the gel moves and all the little rocks move, and that is going to move the cilia and depolarize the cells, and that's how we're going to sense to get the message that the head is tilted. So that's pretty cool. The dynamic equilibrium is essentially twirling, so rotation. That's why we have the semicircular canals in different directions. The place where the receptors are located is in the ampulla, and this is again dynamic equilibrium. In this case, the cilia are embedded into this, again, little jelly-like material, and there is liquid, there is the perilymph all around it, so that when we rotate, like twirling, the perilymph pushes on the cupula, which in turn is going to push on the little cilia of the hair cells and activate them. So it's all very mechanical how it happens. A neuronal pathway for equilibrium, again, the cranial nerve is the same, number 8, and then from there the information will be taken to the cerebellum, which is the main organ for equilibrium, the cerebral cortex, because we'll be aware of what we're doing, and actually the nuclei that controls the extrinsic eye muscles, because the eye muscles are going to move the eyeballs as we twirl. So that is basically it for equilibrium. So this is not terribly bad. We have a few minutes. What we'll do next time is go into the endocrine glands. You know what? I'm going to stop the recording and probably